break, comfortable.
Yeah.
Keep an eye on things in here.
Definitely.
All right. Be ready for that current to pick up too. I know you were here yesterday, so you should be good.
I'll keep an eye on it.
So if you've missed any of the days of the expedition, you can visit the website, oceanexplorer.noaa.gov, Okeanos. Then go to the Explorations EX1811. And there you'll find a bunch of information, the link to the live videos, some of the daily updates, mission logs. And then you could go to the image gallery to see some of the photos taken and highlight videos.
Pretty significant.
Produced by some of our very talented videographers.
Yep.
Also, there's a link for educators, so you could
curve?
find classroom lessons, both in
Still
English and
similar?
Spanish.
Probably not terribly well.
And some of the learning tools that are relevant to this expedition.
Like 45.
And these are separated by grade level.
Significantly higher.
Copy that.
Oh, yeah.
Pilot. Yeah.
Co-pilot, how are you today?
Good.
Yeah, I just died there as well. A little bit.
If you want to practice flying against it, I mean, feel free.
Yeah, I think I'm gonna do that.
Okay.
I've been seeing a lot of sea urchin spines.
Copy that.
Yeah, it'll be good to take a look and see sonar. How it looks.
Pilot, that larger white fan in the distance, can we take a look at that for associates?
Distance as far as
The
11 o'clock
yeah,
or like 2?
11 o'clock, 12 o'clock, yeah.
Oh, here's
Large
one.
white fan. Oh, that's a
Is that-
Stichopathes.
I'll get you move after you.
Very large
so
one,
you have
yeah.
plenty of time.
Copy that.
Just be mindful of this guy.
Yeah. Trying to go up and over him.
Nice.
I think I dodged. Well, actually, just let me come around here.
Okay.
Line up with the current a little bit better.
It's just I noticed it had a lot of associates on it. It might be worthwhile to take a closer peek.
Copy that.
I think in terms of numbers of associates, Calagorgia has by far surpassed any of the others
You can
corals we've seen on this dive.
You got the tether.
We saw a very large basket star earlier on another colony. It's not uncommon to have many possibly different species of brittle stars on one octocoral colony, or maybe a crab, chirostylid, or squat lobster.
Come in.
I think we're just gonna have a few different crinoids here. These look very much like the crinometra
Come
species
over to the
that we
port
were
a
looking
little
at
bit.
earlier.
Holding there.
One of the branches, actually the far left branch, looks like it was lopped off a bit.
Okay, coming to the
Yeah, it looks
far left.
chopped.
The top too. Huh?
Mm-hmm.
So we're just taking a look at the diversity of different ophiuroids here.
There's a squat lobster right underneath the crinoid.
Oh yeah.
He's hiding really well.
Okay. Tilting down.
He has
video.
protection from both sides.
Any interest in the base watch leads?
Yeah. Can we actually go back up to look at the squat lobster that was at the top?
Yep, coming up.
A lot of branch loss on the lower end of this colony.
You're gonna have to direct me to. Oh, I see.
Upper left.
Yeah, gotcha. Coming over. coming up. Coming up. That's what I meant to do. Okay.
This looks very similar to that smaller one we sampled a few days ago
Just a tad video. Let me re-get
on
my
the purple coral.
Alright. Jesus, current is
That's all right.
really.
Don't worry about it, pilot. We can move on.
Copy that. Video, I'm assuming you're clear.
Okay,
Thanks.
I'm gonna get you another move in.
Copy that.
Don't sneak too far out there. Bridge, RV nav. Ready for another move. Same move, 20 meters at 090, 0.2 knots. Good copy.
So this expedition is one of several expeditions that will be taking place between now and 2020 that would contribute directly to the ASPIRE program, which stands for Atlantic Seafloor Partnership for Integrated Research and Exploration. ASPIRE is a multi-year and multinational collaborative field program, and it's focused in that's a sponge. And it's focused in raising collective knowledge and understanding of the greater Atlantic region. That includes the Caribbean.
We had a great opportunity to speak with a workshop of scientists and folks who are involved with the ASPIRE program firsthand yesterday. They were holding a workshop to evaluate the different proposed targeted areas for the ASPIRE campaign and understand which areas of research might best suit those priorities. For example, geographic locations or topics. So we spoke yesterday with about 60 or 70 folks at the workshop, some of whom may still be tuning in today. I think that runs through today. I don't think we've done our round of afternoon introductions yet. We'll start in the back row, I suppose.
Move here when it's done, okay?
My name is Steve Oskovich. I'm one of the co-science leads on this expedition to explore the deep water habitats around Puerto Rico and the U.S. Virgin Islands.
Is that a fish there?
So
He's not going to get away.
I'm a PhD candidate at Temple University in the Department of Biology, Philadelphia, Pennsylvania.
Feel free to zoom in on
I
this little
am
guy here.
a deep sea coral ecologist focusing
Yeah,
on
green eye.
distribution and biogeographic patterns
Yeah.
of deep water
green.
corals.
Feel free to come out. Just curious.
Small little fish.
Yeah, green eye. It's a bunch.
Oh, there's another one.
Thank you, Bridge. We'll go ahead and put another one in right now then.
Could we snap zoom
20
to the right
meters,
of the
090,
lasers?
0.2 knots.
Okay. Great. This thing coming up.
Yeah.
Good, Captain.
Alright, feel free to come in video.
It's just a crinoid on a sponge. Oh, okay, thank you. There's some cup corals. There's that sponges.
They're everywhere.
There's-
If you look close enough, you'll find them.
Free to come out video.
And my name is Stacy Williams. I'm the other co-science lead on this expedition. I live in Puerto Rico, and I'm a co-founder of a nonprofit organization, the Institute for Socio-Ecological Research, and also for a for-profit organization, Coastal Survey Solutions. I'm a benthic ecologist, and I've been surveying different marine communities, seagrass to shallow and deep water habitats around Puerto Rico and throughout the Caribbean.
Any interest in this science? Similar to what we have been looking at?
We could take a quick snap,
Okay.
just to make sure it's the same.
Come in video. Yeah, the stem looks the same. Recognize that?
You know, very,
Yeah, the nodes.
very heavily.
Come in. 1 second.
Very heavy protein buildup on those nodes. Those black nodes we're talking about are made of a protein called gorgonin. It may give some flexibility to the bamboo coral. This one has a really bizarre branching pattern. The branches are coming
Go
out
ahead and come in.
pretty much all in the same spot.
Come
Hey,
down.
Steve, it's Scott France here. I agree. This is a pretty unusual nodal structure there at the base. They almost seem to be bulging out like herniated discs. And the unusual branching structure you're referring to, I don't know if that is a normal for whatever taxon we're looking at here, or if it's the result of having some injury to the tissue and regrowth gives it that odd pattern. But this is certainly very distinct.
Yeah, very interesting for sure. It's not something we've seen so far. Do you have a thought about which clade it could belong to? We were noticing before that these colonies were branching just above the node in many cases.
Yeah, hard to say. If pushed, I would say D2 clade. So that would be the formal keratoisis clade. And there's a couple of things, I'm trying to remember the names now, gray eye and maybe flexibilis that could fit. But that odd, cluster of branches where it arises is not something I recognize.
Yeah. Yeah, we did notice that there are some anemones that were observed on
Fetching it
another
from
colony. They
Move
seem
should
to
be
have
hitting, so I think you're fine.
caused,
Copy
actually I
that.
can
Thank
see
you
one in
for
the upper
that.
left-hand corner too. They seem to have caused some tissue loss in the vicinity of where the anemone was attached. So it could be associated with those sorts of
Coming up to that
injuries. But
little,
thanks, pilot. We can head on here.
copy that. Video, you clear? Okay, let's continue. Lasers back on, please, co-pilot.
We tried to get a good image of the polyps previously too, and it's sadly not a lot of contrast here against the soft bottom, so they're pretty translucent.
Thank you, Bridge. watch your toes there.
Yeah. Notice that. Been kind of weaving in and out of a lot of them. Like green eye. Feel free to call out any fish you may see in the Sirius cam.
Yeah, I haven't seen too much yet.
Yeah, 'cause like now the current's pretty much dead. It's real weird fluctuations.
There's something branching right behind these pairs of crinoids.
Copy that.
It's at 11.
Coming back.
It's
to
like
it.
one single branch. I don't know if it's those suanthids.
Co-pilot, could you cycle his aft cam, please?
Are you talking like
If
10
you go
o'clock
like,
right now or
yeah,
12?
like 11
details.
o'clock or 10.
Yes.
Yeah,
Coming up now. It's in between the lasers.
yeah,
Alright.
that.
Perfect. Thank you.
Copy that. Just a snap zoom video. Okay. Do you want to get closer to that?
Oh yeah.
I just need to maneuver through some of these other crinoids first.
Yeah, if you can, that is
Yeah.
an interesting coloration pattern.
Of course. Video come out.
You know, I was just wondering, we're seeing yellow and purple on there. I wonder if this is a missing link that could tell us if there's any similarity in possibly or intra-specific variation in coloration between this purple plexaurid we were seeing and some of these yellow plexaurid fans. We were noting the other day that purple is not an extremely common coloration pattern for a plexaurid, but it also didn't have any other blended colors in there, as we sometimes see blues and greens in a single colony. But this one's not very large, but it may provide us some context about how color is variable in this plexaurid species.
Okay. Come in video. Up. Okay, go ahead. Sorry about that.
You want me to
Drive
do
the
your
forward.
Forward instead of down.
Yep.
take your rotated?
No.
Okay.
Thank you though. Yep.
In this kind of current, you're going to need more down than you are forward the way it's set up right this moment.
Copy that.
There you go. That's looking good. Now maybe just a little bit
This
forward.
is if this is the same kind of purple plexaurid stalk, I've seen another one today, but this one is much larger and actually has branches, whereas the other one was kind of just a single stem.
Yeah, it looks half purple, half yellow.
Yeah, right?
Because this is purple, and then there's yellow.
Can we, once we're settled, can we just go from bottom to top, or top to bottom, and just kind of scan through this colony?
Copy that.
Field lights may or may not help on this one.
Yeah, if you want to try and do the FR, make sure he knows that they're coming on though. Okay, I'm going down to the base first.
So
Coming on.
kind of a woody looking stem or base. Actually looks like it's on that sponge, although I can't tell if that sponge is just growing around it or
Coming.
or what. So purple kind of transitions to yellow or golden.
Alright, there we go. Okay, coming up.
A hybrid.
All the way up.
The stem remains purple. And then the branchlets up here turn golden yellow.
Continuing up. Top. If you want, I can go back down. I can go down smoother than I can go up, I believe, in my current position. So, heading back down again.
Yeah, very interesting. I'll have to compare this to what the other colony looked like. Okay, thanks, pilot.
Problem. Video, you happy?
There's a fish right above you there. That's a big one.
Oh,
If you look
big
up,
fish in the distance.
right. He just swam back out of the light pool, but he was a good size. He swam off to the left.
Okay. Just gonna come up a little bit, swing over there, see if he's still there.
Okay, you got a little bit because we're stopped, so
Yeah.
you have a little bit of tether.
Copy that.
Shall I kill the...
Yeah, if you want to come off with the headlights, ask video first though.
Yep.
And then there's
Coming up
something
now.
that's weird structured right there. We might want to get a zoom before we go
The bowl-shaped coral?
past it. You see that black outline of the square?
Oh, yeah. Oh, there's a
Ah,
fish.
there's a, yeah, right there in the water column.
Get a zoom before we go
The bowl-shaped coral?
past it. You see that black outline of the square?
Oh, yeah. Oh, there's a
Ah,
fish.
there's a, yeah, right there in the water column.
Squeezed together so you can't see it amidst the tentacles.
Yeah. Possibly in the genus Liponema, which are the pom-pom anemones, but could be something different too.
Moving on.
Usually the pom-pom anemones have a kind of
So you're still at 0,
ball up
0,
like a
down. Yeah, it's
cluster of tentacles
bouncing around.
protecting themselves on all sides.
Basically 85 or so. Yep, there we go. It's right about 90. All right. Probably do another move. Yeah, I'm ready.
Zulu Charlie.
Bridge, this is ROV now.
Pilot, this is
like
watch lead.
Range,
Go ahead.
20 meters.
As much as
0,
possible,
90 degrees, speed decimal
you know,
2.
can we try and keep the camera pointed up? I think I don't notice too much,
There's the small.
but like periodically
Gopper Bridge, thanks.
tilt up the camera so we can see out into the halo of light to see for the fishes.
Sure. Yeah, we're keeping an eye on Sirius too.
Great.
That. Can see further. There's a little fish. Any interest in this silverfish?
I think it's just one of those green eyes. We
Okay.
could do a snap zoom.
All right, quick zoom here, video.
Maybe not. Looks a little silvery. I know the green eyes are pretty silvery, but this one looks a little bit different. Maybe a juvenile something. The wash on the backside, or at least the way that the light reflects off the scales, seems to be slightly greenish and silvery on the sides. A dark splotch on the dorsal fin.
All right, we can let him go.
Yeah.
Got another one here. Yeah.
Yeah, that
go
seems a little bit different. Maybe what was it, Epigonus?
Could be.
So that's the green eye. Okay, we can go what?
I didn't notice the second dorsal fin.
Yeah.
I think I just noticed the one, and then they had a dorsal fin much further back. So these commercially fished snappers, this brings up an interesting point. Do they have do their juvenile stages, are they deep water juveniles or shallow water, and they migrate to the depths, or do we know anything about that?
Well, they know much more about the silk snappers. So they do have an ontogenetic shift in when they mature. So their juveniles are closer to shore, and then when they mature, they move to deeper waters. They suspect maybe the queens do the same, but I don't think it's too clear.
How's the surface current? It's picked up a bit.
It's not too bad. I mean,
yeah, it was down to 0 like maybe an hour ago.
Really?
Yeah.
We got decimal 3. Let's get you at 40. About ready for another move?
Yeah, I'm ready.
Probably hasn't. Last one hasn't finished yet, but has it? Or
Yeah.
Just about to.
I think it did. Maybe not.
Is that what that rumble is?
Yeah, I feel like it's still.
Pilot, actually,
maybe
can
it's
we
better to-
take another closer look at this silvery fish? It's at 3 o'clock right now. It's out of the screen, I believe.
Yeah, there we go. Now it's spinning.
I think this is, I'm not sure if this is the green eye or if it's the
Oh,
other.
I see it.
Yeah.
Down lower right here.
It's almost it's like almost above the lasers now.
Yeah, I see that one.
Yeah.
A bit a little closer.
Or is this the green eye? This one is the green eye. Yeah, this is not the
Yeah.
one that we...
Okay. Come wide.
Move complete. Thanks, Bridge.
Yeah, that silvery one was very silver from a distance. We'll keep an eye out for it. So that one's done.
Yep. They just finished.
So my question
Bring
is,
another one
why weren't there any crinoids
is already now.
yesterday?
Yeah, right?
I don't think I saw one crinoid yesterday. Or was
Yeah,
it
like a
yesterday
move,
or the
same as
yeah?
last.
Yeah,
Range
yesterday.
20 meters, bearing 090.
Oh no, it's crinoid. Okay. Thought I saw something weird on the floor. Anyway, moving on.
Good copy. Thanks, Bridge.
Yeah, I don't know if I saw many crinoids yesterday. That's an interesting observation. I don't know if I actually even wrote any down. I would assume that it maybe has something to do with.
Has the current dropped for you?
Yeah, I just
Yeah,
saw
that's a
your
good question.
thermometers were down.
Yeah.
Actually, don't know. I don't even have a guess at this point. The current was perfect.
Heading a lot better.
The substrate was
Something
hard and rocky.
Oh, up in the water.
Yeah.
They love
Show
overhangs.
in video.
Yeah, it seemed to be dominated yesterday by attached organisms and not so much the mobile fauna.
Going away, I think.
Oh, there's a boarfish.
Oh yeah.
Oh, there are two.
Yeah, another one stationary.
Yeah. Oh, we're headed that way. Go wide.
Oh, and there's the current again.
Yeah, I think the current's fluctuating. Even during zoom self-set down, have 30% ahead and down, and then all of a sudden start moving forward.
It's annoying.
Saw it in the last watch too.
For folks on the science party, we are on our way to waypoint 4. I think we're going to make waypoint 4 well in advance of the end of the dive, but we'll keep you posted.
Pilot, this is Watch Lead.
Go ahead, watch lead.
Is this a good time to do a set of introductions?
Oh, sure. Well, we just had a watch change in the first row. I'm Carmel Kletchy, piloting Deep Discoverer. I'm an engineer with the Global Foundation for Ocean Exploration, or GFOE. On my left,
City Navigator, Levi Unima.
And at co-pilot is Tony Arnold.
Over on the right on video, Bob Knott.
we hope everyone's enjoying the dive today.
No. So one of the fishermen,
Is that the boarfish moving along the bed?
yeah. So one of the fishermen mentioned how we were talking about this yesterday, that the queen snappers, when there are boarfish around, they don't like to come, I guess, around the boarfish because the boarfish will not allow the queen snappers to feed.
They look terrifying.
They look very terrifying.
I wonder how you would know that. I mean, unless they had... I wonder how shallow you could see these boarfish and queen snappers interacting.
This one looks like it wants to have a stripe along the side. It's kind of like a mix.
Yeah. Could be. It's probably trying to keep station in the current, too.
Catching up to you, but I think we're finishing up, so should be fine. Yeah.
At some of these depths in the Pacific, we would come across organisms. We would come across a type of, I suppose it's a boarfish relative, but it's called a false boarfish, Neositis. And And those fishes would, you could find them down in excess of 1,000 meters, and they would often just follow along the vehicle and photobomb you at the most inopportune moment, right in the middle of the shot. Quite abundant.
Oh, there's another crinoid moving.
Oh, wow, there it goes.
Oh.
That one kind of tripped and fell there.
Oh, is it going to move with its arm?
Tumbleweeds.
Bioboxes are full, pilots.
I guess that's another way to get around.
Tumbleweed method.
Can we look closer at that? See if it's actually a controlled motion, or is it just using the current?
Has the current picked up again, pilot? Yeah. So this is the second time today that we've actually watched a crinoid kind of crawl while this one's tumbling.
.75, and mostly centered on 90. Oh, fish.
Oh, there's a snapper. It's a queen snapper.
Yep, lasers are on. Yeah, it's going off that one.
Just bombing on through. Yeah. Move complete. Thanks, Bridge.
Is that, I'm curious, is that urchin on the seafloor, is that eating a part of the branch of the bamboo coral? I thought I saw one of the branches might have been on its, underneath the urchin here. Might be a good observation to make. It might have just been my perspective too. I think that's one of the larger of these Histosideris.
It looks like it's like has a haircut. Like all the spines on top are very short.
Something else in there.
It looks like it's kind of eating a crinoid. It looks like a feathery, yeah, like a small crinoid.
Oh, really? Can we get some better imagery from the side? That would be...
Wow, these are vicious.
that would be the...